তো আমরা কি স্ক্রিনটা দেখতে পাচ্ছি? ক্যান উই সি দ্য স্ক্রিন?
হ্যাঁ
হ্যাঁ,
স্যার।
আচ্ছা, স্ক্রিনটা মুভ করছে? আরে
করছে।
স্ক্রিনটা একটু মুভ করছে?
হ্যাঁ হ্যাঁ করছে।
ব্যাস, তাহলে উই আর অল সেট টু গো। ওকে, সো টুডে উই উইল বি টকিং অ্যাবাউট প্রোটিন লোকালাইজেশন ইন ডিফারেন্ট অর্গানেলস। ঠিক আছে? এইখানে বেশি কেউ ইয়ে টিয়ে চালিও না, পেনের দাগ চলে আসছে স্ক্রিনের ওপর। কেউ কিছু চালিও টালিও না। মেনলি আই ট্রাই টু গিভ দিস লেকচার ইন ইংলিশ, কারণ আমি again, who is doing all this? Okay, so mainly I'll try to give this lecture in English because if I have to post it, it will be reachable to many more. So how last time in class, many of you asked me this question, I explained to some say five odd people in Jiagan station also, that how actually protein localization takes place. So today I'll explain this. It's a very nice topic. First, let me complete. Then, if you have any question, then only ask me. কিন্তু ওই মাঝে মাঝে স্যার এটা কি করে হলো, ফার্স্ট লেট মি কমপ্লিট দ্য এন্টায়ার স্টোরি। সো in most of the proteins, almost in all proteins, there is a signal sequence. This signal sequence, it's usually N-terminal, except for your peroxisomes. And this signal sequence basically determines the localization of the protein in different organelles. This signal sequence could be 15 to 30 amino acids. Now, the point is that we are saying that the signal sequence is only significant in localization of the amino acid, but this statement may not be completely true. Why? Because this signal sequence is also very, very, very important for the expression of the protein. If it is an N-terminal signal sequence, it is absolutely essential for the expression of the protein. Also, the level of expression might depend upon the signal sequence. তার মানে হচ্ছে, if you remove the signal sequence, the protein may not express properly. Okay, so it's basically hydrophobic. It could be an alpha helix. Okay, but these are some of the structures. Exact details you will be getting when we move downwards. Okay, so দেখো প্রত্যেক অর্গানেল, ফর ইচ অফ দ্য অর্গানেলস ইউ হ্যাভ আ ডিফারেন্ট কাইন্ড অফ সিগন্যাল সিকোয়েন্স, and whatever we have focused till now is the transportation of protein through endoplasmic reticulum. But it is not like that a protein will get transported through endoplasmic reticulum only. It may get synthesized in free ribosomes and might get transported to nucleus, mitochondria, chloroplast, etc. or peroxisome. We will learn about that as well. But first, let us start with the transport of protein to endoplasmic reticulum first, then to Golgi and other organelles like lysosome, or it could be to different plasma membrane, etc. Okay, so as we could see that for endoplasmic reticulum, this signal sequence is typically hydrophobic. It forms an alpha helix. Okay, that's recognized by the cellular transport machinery. Now, as the translation begins, remember the signal sequence for ER proteins is in the N-terminal. So, as the translation begins and this signal sequence starts to unfold, not in the real sense of the term, হ্যাঁ মানে সত্যি সত্যি আনফোল্ড করছে তা না, starts to unfold মানে যে বেরিয়ে আসছে from the ribosome, then what happens, this signal recognition particle, this is a ribonucleoprotein complex. This recognizes the signal sequence. Now, this SRP pauses translation. Why? Because we don't want the protein, the nascent peptide with the signal sequence to get prematurely folded. If that happens, there is a high chance of মিসলোকালাইজেশন অফ দ্য প্রোটিন, মানে ভুলভাল জায়গায় চলে যেতে পারে যদি ওটা প্রিম্যাচিওরলি মানে সময়ের আগে ফোল্ড হয়ে যায়। তাই জন্য ইটস বেটার যে আস্তে আস্তে স্লোলি when it comes out of the ribosome, its translation is stalled by the signal recognition particle. Now what happens? Now there is a complex form. What is that? That is the signal recognition particle, the ribosome, and the nascent polypeptide chain. So this complex is recognized by the signal SRP receptor, that is recognition particle receptor, which is located on the endoplasmic reticulum membrane. তার মানে হচ্ছে SRP, signal recognition signal recognition particle, ribosome, and the nascent polypeptide. All of these they form a complex. This complex is recognized by the SRP receptor on the rough endoplasmic reticulum membrane. এইবার কি হচ্ছে, the ribosome gets aligned to a translocon. This translocon, mainly sex61, why it's kind of a channel যেটা থ্রু কিন্তু প্রোটিনটা ইয়ার লুমেন এ এন্টার করবে বা ইয়ার মেমব্রেনে ইনসার্টেড হবে। Okay, so once the ribosome is aligned to the channel, now again the post translation begins. তারপর কি হচ্ছে, now the protein enters the through the translocon, it enters the endoplasmic reticulum, and now the signal sequence is cleaved by an enzyme known as the signal peptidase. ঠিক আছে, now the protein folding takes place with the help of chaperone like VIP. This I have already told you in the last class as well. ঠিক আছে, so after entry, হ্যাঁ, proteins follow different paths depending upon their final destination. মানে ER এ ঢোকার পর once it enters the ER, it follows different path. What could be that path? Now here we have to understand something, that for transport from ER to Golgi apparatus, we have this COP2 vesicles, which takes protein from ER to Golgi apparatus. ঠিক আছে? And there is another vesicle, that is the COP1 vesicle, which is for the retrograde transport of the protein from Golgi apparatus to ER. ঠিক আছে, so first let's see the first pathway. It could be from ER if it is a secretory protein, মানে ওর ফেটটা হচ্ছে গিয়ে কোষের বাইরে ওর ক্ষরণ হবে, that is it will be secreted outside the cell. Then from ER it will go to Golgi, then to secretory vesicles, and then to the plasma membrane, and it will be secreted to the extracellular space. If its fate is to get inserted within the plasma membrane. Then from the ER, it will go to the Golgi apparatus. From there, it will go to the vesicles and it will get inserted in the plasma membrane. It could be destined for lysosomes as well. So from here, it will go to the Golgi apparatus, and from there, through endosomes, it will go to the lysosome. Or it may be retained in the ER. প্রোটিনটা কিন্তু ER এর প্রোটিনও হতে পারে। Now for that purpose, it has a C-terminal signal, that is KDEL, short signal. Lysine, aspartic acid, glutamine, leucine. This is a short signal. Okay, so it's not necessary. And also for the transmembrane proteins of ER, we have another signal, that is KKXX. Okay, that is lysine, and then any two amino acids. So what I mean to say is that if that signal is present within a protein, that doesn't necessarily mean that it won't go to Golgi apparatus. Still, it might get transported to Golgi apparatus through COP2 vesicles. However, the presence of this protein means even if it is transported to the Golgi apparatus, it can still be transported back to the endoplasmic reticulum for the purpose of its functionality within the lumen, or it might get inserted in the endoplasmic reticulum membrane as well. So this is kind of a retrograde transport from the Golgi apparatus to the endoplasmic reticulum using COP1 vesicles. Now, the other signal sequences, like I have mentioned, it's not like that all the proteins go to the endoplasmic reticulum. Here are other types of targeting signals as well, like some of the proteins which get synthesized in free ribosomes maybe. Okay, so for them, there is N-terminal amphipathic helix. Recognize, this is for mitochondria. N-terminal amphipathic helix recognized by Tom-Tim complexes. Okay, now for nucleus, there is a nuclear localization signal NLS rich in lysine and arginine. We will study that importins are the protein which are mainly responsible for the transport of this nuclear proteins within the nucleus. Then for peroxisomes, as I have mentioned, there is a C-terminal signal tripeptide SKL, that is serine, lysine, and leucine. Okay, and there is another peroxisomal transported signal as well. So this is PTS1, another is PTS2. I'll explain that as well. For chloroplast, you have an N-terminal transit peptide. Okay, that is recognized by toxic complexes in the chloroplast. So why does this matter? Because misrouting of proteins can lead to diseases like cystic fibrosis, যখন মেমব্রেনে ঠিকঠাক ভাবে পৌঁছয় না সিএফটিআর প্রোটিনটা, cystic fibrosis transmembrane receptor, when it is not properly, when it doesn't get localized within the membrane, it causes cystic fibrosis. Or there could be Alzheimer's due to misfolding of the proteins and their aggregation, and certain cancers. Also, biotechnology use engineered signal sequences to direct recombinant proteins for secretion or for membrane display. Okay, so here we see that these proteins, again, the concept comes of the targeting signals. So the proteins that may get synthesized in free ribosomes or ribosomes bound to, yeah, that is in rough endoplasmic reticulum, right? So important thing is these signals are recognized by receptor proteins, like in case of ER, SRP receptor or transport complexes that guide proteins to its destination. Now, this I have already mentioned, so this is just a detailed one. You can take a look at it. Yeah, so the same thing for NLS, we have nuclear pores and importins, mitochondria, Tom-Tim complexes, chloroplast, we have toxic complexes, peroxisomes. For peroxisomes, we have PEX receptors. And for definitely for ER, we have co-translational import by signal recognition particle and Sec61 transloper. Ever once it reaches the ear for Golgi lysosome and plasma membrane, no new signal, but that is not completely true because for lysosome there is mannose 6 phosphate binding. I'll explain that as well. So, but basically from Golgi, from ER to Golgi, it is taken by COP2 vesicles, and from Golgi to ER by COP1 vesicles, and from there to the membrane by different vesicles, or to the lysosome by endosomes from Golgi. This is how it works. Okay, so let's we have taken the example of a ER based protein. এই ER এর থেকে কি হচ্ছে না হচ্ছে। Now let us see, if it is not ER based, it directly goes to the mitochondria. So for that, the protein is made in free ribosome, and if it is made in free ribosome, now there is an N-terminal mitochondrial targeting sequence, as we could see here, that is a amphipathic helix, other than the not the hydrophobic sequence that we see in case of ER proteins. And this amphipathic helix is recognized by Tom complexes on the outer membrane. Huh? And then the protein is unfolded and threaded through the Tom-Tim complex into the matrix, because the folded protein cannot go through these complexes. The pore size is not that big, so it has to be unfolded. In the mitochondrial matrix, there is mitochondrial peptidases which cleaves the signal sequence. And finally, there are chaperones within the mitochondrial matrix which helps in protein folding. So, there are certain diseases other than this cystic fibrosis and this cystic fibrosis, and the other one, the neural disease that I have just mentioned, Alzheimer's. Okay. So other than that, this dual vigor syndrome, this is called by defective peroxisomal import as well. So this I will skip. Just I have given you more and more of the same information from different AI tools যাতে এটা তোমাদের কাছে একটু ব্যাপারটা ক্লিয়ার হয়। ওকে। সো here I think, yeah. So this peroxisomal, as I was talking about PTS2, now PTS2 is an N-terminal nonapeptide signal. Okay. So like PTS1, which is just a tripeptide signal at the C-terminal, PTS2 is a nonapeptide signal. And again, these are recognized by the PEX complexes. এবার আমরা যদি ইয়ার টার্গেটিং এর ব্যাপারটা আরেকটু ডিটেলসে দেখতে যাই, সো হোয়াট ডু উই হ্যাভ এন রিজিয়ন উইচ ইজ পজিটিভলি চার্জড অ্যামিনো অ্যাসিডস। ঠিক আছে, আমরা তিনটে রিজিয়ন দেখতে পাই। সো ইয়ার সিগন্যাল, এন্ডোপ্লাজমিক রেটিকুলাম সিগন্যাল হ্যাভ আ ট্রাইপার্টাইট স্ট্রাকচার. So in the N region, we have positively charged amino acid like lysine, arginine. In the H region, that is the second region, we have hydrophobic core of 10 to 15 residues, often forming an alpha helix. And in the C region, there is polar residues near the cleavage site. Cleavage site means where the signal peptide is cleaved by the signal peptidase enzyme. Because after the protein reaches its destination, it is imperative that the signal peptide have to be cleaved. So, in general, we could see that there are signal patches and linear signals. So, if there are contiguous amino acid sequences, like in nuclear localization signals, it is called a linear signal. However, there are signal patches as well. What happens? There are different patches in the proteins which come close to each other in the folded protein, forming a particular functional domain. Okay, discontinuous residues যেগুলো হয়তো পাশাপাশি নেই, they are not close to each other, but once the protein is folded they form a functional targeting domain. এগুলো কিন্তু লাইসোজাইমের এনজাইমসে, প্রচুর লাইসোজোমাল এনজাইমসে দেখা যায়। ঠিক আছে, so these things we have already mentioned, and one very important thing, that is many targeting signals, especially the N-terminal ones, share structural similarity across organelles. Means different organelles like mitochondria, nucleus, chloroplast, even ER, all the N-terminal sequence have some kind of structural similarity. Some may have a hydrophobic helix, some may have a amphipathic helix, but there is a structural similarity. এটা কিন্তু this is indicating towards the evolutionary conservation between different mechanisms of transport to different organelles. So this is all that we have for the signal sequence. এটা, though it's not directly linked to your syllabus, but since you have many questions, that's why I have answered. One more thing for lysosome. ওকে, দিস ম্যানো 6-phosphate ট্যাগ, আমি একটা জায়গায় বলেছিলাম it's not completely true that no other tag is needed. So that's where I have mentioned that for lysosome, a mano 6-phosphate tag is added to the protein in the Golgi apparatus itself. তার মানে কি হলো, in the endoplasm, when it enters the lumen of the endoplasmic reticulum, signal peptidase cleaves its signal peptide. Now, by COP2 vesicles, these proteins are transported to Golgi. In the Golgi apparatus, a mano 6-phosphate tag is added to the protein. Now, by means of endosomes, these proteins are transported to the lysosome. এটাও দেখা গেছে লাইসোজোমের প্রোটিনে কিন্তু ডিফারেন্ট প্যাচেস আছে, নট লাইক দ্য এন টার্মিনাল সিগন্যাল পেপটাইড. there are different patches যখন সেই প্রোটিনটা ফোল্ড করলো গলজিতে, then there is a structure formed, three-dimensional structure formed by these patches, which itself is a signal for localization to lysosome. It happens in case of lysosomal enzymes. However, this mannose 6 phosphate tag, which is added to the protein in the Golgi apparatus, now there are mannose 6 phosphate receptors available. Okay, so this aids the protein in its delivery to the lysosome. So এটা নিয়ে আর দীর্ঘায়িত আমি করব না। Yeah, so stop sharing. Pause.